Now, perhaps you have reason to be nervous, pun intended, that we are studying the nervous system, because it is actually kind of the most abstract. Most of the physiology that goes on with the nervous system you can't really see. It's happening at the cellular level or the chemical level. And so it can often be very complex. But a lot of the things that we'll be studying with the nervous system, we have already touched on when we talked about the muscle system, when we talked about cells. And so hopefully this will really be bringing things together that we have already learned and then adding some detail on top of that. This is also the last system that we're going to study for A&P 1. We're going to spend the most time on it because there's such a complex array of anatomy and physiology involved with it. But this is it. It's also very central to understanding the systems in A&P 2. So it is something you're going to want to make sure you know and know well. But the main function, a scenario that I might put forth to you, is, let's say you are sitting outside enjoying a nice barbecue, or excuse me, in the South it would be a nice gathering where you are grilling food. Let's say you're sitting there and somebody's throwing or kicking the ball in the yard, and then that ball makes its way towards you. Okay, this is where the nervous system is always working, but this is kind of where it kind of comes to front. You are going to be gathering information from the world around you, your external environment, through sensory receptors that you have, including sound waves being perceived in your ears, your sense of smell, your sight, even your awareness of the people around you. All of this is kind of the first part of the nervous system, gathering sensory information, and then your brain is going to try and make sense of all this. So it's going to integrate. Okay, people are looking worried as this ball is coming towards me. I may be able to hear it. Somebody may have shouted something, and then you're going to have a response to that. So response may be to duck and hold your head, or to turn towards where you think it's coming from. And hopefully that will end in a safe encounter. But every once in a while you may actually experience more sensory information from the external environment as the ball hits you in the face. So again, those three major functions of the nervous system are integration, thinking and processing, which occurs generally after you have taken in a bunch of sensory information. And then your response to that would be motor movement. And that's simply the functions of the nervous system. However, the actual way in which it plays out is very complex. All right, so we're going to start by looking very small at the anatomy of neurons. Okay, the neurons have a cell body consisting of a nucleus, cytoplasm, and organelles, just like every other cell. A cluster of cell bodies is called a nuclei in the central nervous system and a ganglia in the peripheral nervous system, and they're generally gray in matter. Okay, and so you can see some here, this nucleus found in the brain. And then there are neuronal processes, so things coming off of the cell body in different directions, generally in two directions. A branched region is called the dendrites, and then the part going away from the cell body is generally called the axon. So you can see that here. Here's our cell body with our organelles and nucleus. Here are the dendrites, and here's the axon moving away. There is one axon per neuron, and the parts of it can also be divided into the axon hillock, this area which is kind of triangular in shape. The long axon is called a nerve fiber. So this may stretch for a number of a certain distance depending on the type of neuron that it is. Branches of axons, so coming off of here, you may have an axon collateral, and then you have little branches at the end called terminal branches. So there's no axon collateral on this one, but here are some terminal branches. The very distal end is called the axon terminal, and that is where a synapse or the signal will be passed to the next neuron or whatever tissue it's innervating. And that signal is passed through chemical messengers called neurotransmitters, which we've come across before. All right, surrounding this whole cell is the axolemma, which is simply the plasma membrane of neurons, so a specific type of plasma membrane. And it may also have external support tissues, cellular products called the myelin sheath. This will protect and insulate the axon to allow the conduction speed to increase. There are different types of neurons, okay, 3 different types in the nervous system. Unipolar neurons, these generally only carry sensory information, and this is how they are shaped, with the axon splitting and the cell body in between that split. Bipolar, these are rare, but this would be very in the middle of the axon, and these generally just carry vision for sensory information such as vision, hearing, taste, and smell. And then the third type, which is the most abundant, is multipolar, which are motor and interneurons, so those that are actually innervating the target tissue and the ones in between them. Interneurons are then association neurons, so middlemen that occur between the sensory and motor neurons in the central nervous system. There are many support cells of the neurons, cells surrounding them that have helper functions but are not actually neurons themselves. They're called neuroglia or glial cells, and there are 6 different types. 2 are found in the peripheral nervous system and 4 in the central nervous system. The 2 in the peripheral nervous system are satellite cells and Schwann cells. Satellite cells wrap around the cell bodies and protect essentially the cell body from what could be damaging in the blood. So not everything in the blood, the neurons don't want to be exposed to everything in the blood because of their importance, and so there's a filter there essentially formed by the satellite cells. And then Schwann cells myelinate. They produce that myelin sheath, which allows for faster transmission of the speed of the signal. And they are white in color nerves, which are extensions of axons, white in color because of the Schwann cells and the myelin sheaths around them. Oligodendrocytes basically do the same thing as Schwann cells, just in the central nervous system rather than the peripheral nervous system. And oligodendrocytes can myelinate many processes. So you see this one here, even with only one oligodendrocyte. Astrocytes form the blood-brain barrier. So again, like the satellite cells, They prevent blood from actually fully interacting with the neurons and mediate that exchange. We have microglia. These are phagocytic cells, so these are immune cells which are important for preventing infection and or recycling old and damaged cells. And lastly, ependymal cells, which are important. These are ciliated for and line the areas where cerebrospinal fluid congregates, which is in the ventricles of the brain and the central canal of the spinal cord. So they help circulate the cerebrospinal fluid, which is essentially the nutrient fluid of the nervous system. All right, so myelination occurs through a three-step process. The three steps are where first the Schwann cell surrounds the axon, okay, and then it just kind of wraps around and around and around, kind of like you would like saran wrap, and creating these multiple layers, and eventually then the cytoplasm is squeezed out of that, and that then adds that insulative myelin sheath layer there. Okay, and you can see that in this micrograph, all the layers created by that cytoplasmic squishing of wrapped around myelin. All right, how does then the myelin sheath help signal transmission? Well, it does it because by insulating the signal, it makes it so the signal does not have to recharge at every step. It only has to recharge at these nodes, areas where the myelin does not cover it, called nodes of Ranvier. Because then it only has to recharge at those points, the signal goes much faster. Whereas one that's unmyelinated has to recharge every point along the way, and so that's a little bit slower. All right, so we mentioned nuclei in the central nervous system, which is a congregation of cell bodies which don't have much dendrites or axons. They're called ganglia in the peripheral nervous system, and these are essentially unmyelinated neurons, which are important for communicating and processing information and not transmitting information, which is what nerves and tracts do, and these are found in the peripheral and central nervous system. And they are very myelinated. They aren't concerned as much with integrating and understanding or making sense of the information, but instead just transmitting it. All nerves, tracts and nerves look the same. Tracts are myelinated again by oligodendrocytes because they're in the central nervous system, and nerves are myelinated by the Schwann cells in the peripheral nervous system. All of these nervous tissue has layers of fascia, layers of connective tissue, and we've already looked and seen connective tissue in the muscles and layers in the bones as well. The three layers of fascia are the epineurium, so epi meaning outside, perineurium, this covers fascicles or bundles of nerves, and the endoneurium, which covers then neuronal processes. Schwann cells are underneath the endoneurium, deep to them, and then cell bodies and short processes are unmyelinated or gray in color. Okay, so that's some of the basic anatomy of the nervous system. We're going to now talk about neurophysiology. So this is where it can become pretty abstract and hard to understand, but we've already gone over much of this, including the electrochemical equilibrium of a cell. As you remember, this electrochemical equilibrium is a negative charge inside the cell difference. There are much more negative charge inside the cell, fueled by these large proteins which can't get out of the cell. For neurons, the resting potential is negative 70 millivolts if you remember, muscles were negative 85. And these ions are then in different abundance inside or outside of the cell. Sodium, calcium, chlorine are in greater abundance outside the cell, and potassium and the negative charged proteins are in greater abundance inside the cell. Membrane channels are then what regulate the changes in potential, or the movement of these ions between the plasma membrane inside and outside, then change that potential, change that difference. Leakage channels are always open, so there's always a little bit of those ions associated with those channels going in and out, sodium and potassium generally. Gated channels, open and close in response to some sort of stimulus, and block then those passages of substances. Some of them are chemically gated, where a neurotransmitter opens and closes the gate. Some of them are voltage gated, where a change in membrane potential, an electrical stimulus, opens the gate. Some of them can also be mechanically gated, where actual some sort of physical movement or pressure opens and closes the gate. And so we've talked about the neurotransmitter and the chemically gated channel opening. We've talked about the voltage gated response to an action potential, but also you have physical pressure, which then causes the mechanically gated channels to open. All right, and over here we would have our sodium-potassium pumps and leakage channels, which would also work to maintain that potential. We already talked about this before, but just briefly in review, potassium naturally flows out of the leaky channels, which creates an electroequilibrium of about 90 millivolts. Sodium naturally flows into it, and the sodium-potassium pumps counteract the leaky channels to give you an equilibrium at about negative 70 millivolts in the neurons. And if you remember, that is the point then at which this electrochemical equilibrium is achieved, where the attraction between the positive and negative charges, between positive ions and negatively charged proteins, is balanced by the chemical gradient, or the desire to diffuse from greater to lesser concentration. All right, so now the action potential. All right, so this is a change in membrane potential, and this is how a signal is sent. And there are two parts of it which are important. Depolarization is where the voltage becomes less negative. So we're going from a negative to a positive, or a negative to a much less negative. And then you also have hyperpolarization, where you become more negative. And you can see that here. So if we're started at negative 70, this would be a depolarization event. Hyperpolarization would be something below what we had at rest, which happens at this dip here. The action potential then, this is one. This is what happens over time. And this is talking about inside the cell. Then follows the step. The generation of the action potential has both these D and hyperpolarizations and repolarization. So the resting state, only the leakage channels are open. In depolarization, the voltage-gated sodium channels open, and that causes sodium to rush into the cell. And so the cell becomes much less negative and even positive. In repolarization, the sodium channels are inactivated, and the voltage-gated potassium channels open, and then potassium rushes out of the cell. So now it's becoming more negative. And it becomes so negative that it overshoots then the resting potential, and becomes hyperpolarized. And then to maintain that resting state, restoration occurs, so the sodium-potassium pumps restore that electrochemical equilibrium. This is an all or nothing event, whereas if it starts, it can't stop in the middle and restart, or it just goes through to completion. See, and you can't have a small or large action potential. It's just an action potential. Goes through the same steps every time. The voltage-gated sodium channels then have 3 positions: closed, which is at rest; opened, which is when it's depolarized; and inactivated, which is when it's repolarizing. Okay? The potassium channels just are closed in depolarization or open in and hyperpolarization. All right, so what do these steps then look like when you're looking at our channels? Well, at rest, everything's closed off. You have some leakage channels where some of these are coming in and out. The action potential then starts, and that opens the sodium channel. Sodium rushes into the cell, so now we're becoming more negative. Then the potassium channels open, and the sodium channels are blocked, are inactivated. And so potassium flows out, and we get a decrease in polarization, a decrease in charge, to where it then overshoots. Okay, lots of potassium goes out, hyperpolarization, and then return back to the resting state occurs as sodium-potassium pumps restore those original concentrations. Once an action potential begins, it goes, the change within the cell, it then goes down the cell. It propagates down the neuronal tracks, or the axons. It can't go backwards because of repolarization. And so an action potential that would start in the middle would go in both directions, but generally it just goes from one direction to the other. So if this is the beginning resting potential, this is where a depolarization occurred, and then this is in the next stage, hyperpolarization. So this can't cause this cell and the signal to go this way because of that hyperpolarization, so it only then goes down the axon this way. So the next step would be here and here and here and so forth. All right, so action potentials are an all or nothing event, but graded potentials are not. They are additive, meaning or subtractive. They will, depending on whether it's a positive or negative response, add or negate to each other. And if you have enough of them, they can cause an action potential. There are 2 different ways this can occur. It can occur through spatial summation, where essentially multiple signals at different dendrites will initiate an action potential. So they will go above the threshold as they are added up. In temporal summation, then, it's where an action potential or this graded potential occurs at a frequent enough event, or frequent enough times in a short enough time, that it causes an increase in depolarization and causes an action potential. All right, so what am I saying here? Well, if negative 55 here is the threshold for this action potential, you may have these stimuli at different locations. So a temporal summation would be as if if these 2 went off at the same time, then they would have an additive effect. But you also have inhibitory graded potentials. And inhibitory, if that one goes off, then that's going to decrease. Okay. So over, it looks like these 2 kind of fired right next to each other, and they added. This one then fired and it decreased. We had another one fire, and then finally enough fired where an action potential would occur here. Okay, so an inhibitory postsynaptic potential, an IPSP, then decreases the the voltage so it gets further away from the threshold. And essentially, chemically regulated, potassium channels open and they cause potassium to go out of the cell. So now it's less negative, hyperpolarization. A local current then is where diffusion of ions across the cell body go to the axon hillock, and graded potentials travel by this local current. All right, and again, if enough of them occur, either let's say this one fires twice, 1, 2, and then the inhibitory one fires, they can eventually, it'll stay under here. There will not be an action potential until that threshold is met. Okay? This then is the axon terminal. It looks a lot like the neuromuscular junction, except on the other end you have another neuron. The process is very, very similar. The action potential moves down the neuron into the axon terminal. That causes calcium to go in. Synaptic vessels then release neurotransmitter, and then that causes these sodium-gated channels to open on the other end. All right, there is a period of time after a neuron is stimulated by which it cannot go again, and that's called the absolute refractory period. So if another stimulus occurs during this time, when depolarization is occurring, it doesn't matter. It can't do another action potential at that time. In the relative refractory period, it can, but it has to be a very, very strong stimulus to cause another action potential, and generally most stimuli won't. Okay, so that's it for neurophysiology. Again, very abstract, lots of graphs, things that we're going to explain and go more over in class. But we're going to zoom out right now and then just talk about the divisions of the nervous system real quick. We have been talking about this central nervous system and the peripheral nervous system, but we haven't really defined it yet. The central nervous system is your brain and spinal cord. Okay, this is where most integration occurs in the body, where all sensory information is sent. The peripheral nervous system then are all the nerves and tracts running away from there. Okay. The peripheral nervous system then can be split into many different sections. Okay, it can have sensory information that's going in towards, that's the afferent nervous system, or it can have motor information being sent away, that's efferent nervous system. Exiting I don't know, I don't have a good word for A, but E means exit, so that's how I always remember it. All right, the motor, let's talk about the sensory nervous system. There are a number of neurons going into the brain, including these cranial nerves. Okay, some of them have to do with special senses, such as the first, second, 7th, 8th, 9th, and 10th. We'll go over these more later. And then you also have somatosensory nerves. These are that have to do with body senses rather than special senses. Spinal and cranial nerves that detect touch, pressure, pain, temperature in the joints, skin, and muscles. You also have viscerosensory, which are spinal nerves that detect touch, pressure, cramps, chest pain, and temperature of the visceral organs. So included in the cardiac muscle, smooth muscle, and other organs. All right. The somatic nervous system, or the motor efferent, includes voluntary commands from the central nervous system to the skeletal muscle. So these are the only parts of the body where we can consciously control. And there are a couple times couple of phenomena or conditions where these things get a little mixed up. Paresthesia is where your arm or your hand falls asleep, and that's generally due to pressure on the nerves because of a position or pressure that's being put over a period of time. Another kind of interesting phenomenon which occurs is sleep paralysis. This is where you may have woken up and not been able to move. Generally, when you are in a deep REM sleep, essentially your brain is awake, but your somatic nervous system is still turned off, so you can't move. I don't know if any of you have experienced this, but I have, and it's a little bit freaky. All right, so somatic nervous system is part of the efferent nervous system. The other part is the autonomic nervous system, and this is all involuntary and includes our cardiac and smooth muscle and glands, not our skeletal muscle. We can split the autonomic nervous system into two systems: the sympathetic nervous system, which is stimulatory to speed up the heart rate, breathing, sweating, getting ready to move, essentially. We do this when we are exercising. It's very active, also known as the fight or flight response. So it can be very suddenly turned on, let's say when you are trying to escape some sort of large creature, or when you are scared, or even just exercising and moving your body. The other branch of the autonomic nervous system is the parasympathetic nervous system. This is more inhibitory, so rather than when you're moving, when you are resting. And this slows your heart rate, is important for breathing and sweating. Essentially the rest and digest nervous system. All right, so when we map these all out, this is what it looks like. You have your central and peripheral nervous system. The peripheral nervous system splits into our afferent division, which is information coming in to the central nervous system. And then you have the motor efferent nervous system moving away, which can be split into the somatic and autonomic nervous system, and the autonomic nervous system can be split into our sympathetic and parasympathetic nervous system, and that's it.